Welcome to Biochemistry Basics. Have you ever wondered how tiny molecules come together to create the amazing phenomenon we call life?
In this course, you'll explore the 5 foundations of biochemistry: cellular, chemical, physical, genetic, and evolutionary biology. With clear explanations, stunning visuals, and real-world examples, you'll build a strong foundation in the chemistry of life. Subscribe, and let's begin.
Let's explore the chemistry of life, from water, and discover how molecular structure, interactions, and information drive every biological process.
For centuries, early biologists looking at a living cell faced a daunting epistemological roadblock. They had to ask whether the complex processes animating living things were driven by an unquantifiable life force, or if they were bound by the same universal laws of chemistry that govern the inanimate world. By the late 19th century, the scientific consensus heavily favored a concept known as vitalism. Leading researchers of the era, notably Louis Pasteur, championed the idea that biology operated under its own distinct set of rules. Pasteur observed processes like the fermentation of sugar and concluded they were inextricably linked to the vital processes within living Saccharomyces, or yeast, cells. Under this framework, separating the chemical reactions of life from a living, breathing organism was considered a physical impossibility. This assumption created a hard boundary. It effectively walled off the study of biological organisms from the rigorous, mechanistic analysis available in a traditional chemistry lab. That boundary stood until 1897, when a German researcher named Eduard Buchner executed an experiment that dismantled the Pasteur consensus entirely. Buchner's methodology was brutal but effective. He took ordinary yeast and sheared apart the cells, violently crushing them to extract their internal juices. Crucially, he filtered this extract to guarantee a strict experimental control. Absolutely zero intact Saccharomyces cells remained in the collected liquid. He then introduced sugar to this lifeless liquid. The completely cell-free juice proceeded to ferment the sugar perfectly. A reaction previously thought to require a living organism had been replicated entirely outside of a cell. The vital processes characterizing life were abruptly pulled into the chemist's laboratory to be studied in a test tube. Buchner's discovery provided the foundation for a new discipline, moving the investigation of life out of the realm of philosophy and into the laboratory. The same chemical laws that hold for the test tube apply to the cell, proving that the living state is an ordered chemical entity subject to systematic analysis. To understand how those reactions operate, we have to scale down from the test tube to the atomic level, starting with the basic building blocks of cellular chemistry, monomers. Within any cell, these individual units fall into distinct classes, primarily amino acids, monosaccharides, and nucleotides. Monomers don't drift aimlessly. They are locked tightly together by rigid covalent bonds, which physically force the growing polymer into a stiff, one-dimensional line. But this isn't random. The exact sequence represents highly ordered data, revealing a primary rule: sequence is information. When specific nucleotides are strung together, they form DNA, providing a massive, highly readable molecular blueprint for an entire organism. Even shorter polymer chains, like oligosaccharides composed of sugars, utilize their unique sequences as precise points of molecular recognition for other cells. By locking monomers into a strict order, covalent bonds write a physical script. That linear sequence acts as the foundational code required to build all cellular complexity. This presents an engineering problem. A rigid, one-dimensional string of data cannot perform the physical labor of a cell. It must somehow transform into a dynamic, three-dimensional machine. The solution emerges as these polymers scale up into massive macromolecules, and the governing chemical mechanics shift away from those rigid covalent bonds. While covalent connections successfully dictate the underlying sequence, they lack the flexibility to govern the final biological shape. Instead, the 3D architecture is dictated by aggregate non-covalent interactions. Hundreds of these faint physical attractions pull on the chain simultaneously, overpowering its one-dimensional stiffness. They force the polymer to collapse inward, bending into a highly specific, densely packed 3D conformation. Because so many non-covalent interactions pull at once, they lock the macromolecule into its native state with immense stability. Yet because each interaction is weak, it retains localized flexibility. Without this exact combination of stability and movement, macromolecules would be durable but completely paralyzed. It is this specific folding mechanism that breathes mechanical life into the sequence. The direct relationship between a monomeric sequence and its final folded geometry is most clearly demonstrated in proteins. Every protein obeys a strict operational rule. Its unique three-dimensional structure exclusively dictates its biological function. We see this in the Brie shapes. First, an amino acid sequence folds into an interlocking cable, providing rigid support like hair or wool. Alternatively, a different sequence collapses into a globular sphere with a hollow pocket, perfectly engineered to transport oxygen. Finally, a third sequence folds into a highly dynamic enzyme, shaped precisely to capture other proteins, physically bend them, and cleave their molecular bonds. Whether a protein provides structural reinforcement, internal cellular logistics, or oppressive chemical catalysis, its operational capability is a direct mechanistic manifestation of its physical shape. Scaling up from individual proteins brings us to the final tier of this chemical hierarchy, physical aggregation. Individual fully folded macromolecules do not act in isolation. They begin to aggregate, locking together seamlessly to construct massive, highly functional supramolecular structures. When thousands of lipid molecules aggregate alongside specialized signaling proteins, they physically combine to form a continuous cellular membrane. These immense structures then interact to build the distinct organelles that execute the many metabolic functions of the living cell. This entire cellular machine rests on a strictly logical foundation. Every biological action taken by the complete organism relies entirely on the precise sequencing of covalent bonds at the very bottom of the hierarchy. The regulation of homeostasis is a direct manifestation of these chemical signaling systems, coordinating molecules within cells and across organs to maintain biological stability. This hierarchical architecture, from covalent sequences to complex folds, allows investigation of the living state. Biology is a system understood through its structural logic.
Every great scientist starts with the basics. Subscribe to Biochemistry Basics, like this video, and ring the notification bell to join us for every new lesson. See you next time!